Thank you for joining my poster presentation on an evaluation of censusing methods for Western lowland gorillas. Western lowland gorillas are a critically endangered subspecies of gorilla found in Western equatorial Africa. Because of poaching and disease, their populations have declined over the past quarter century. However, currently the exact number of Western lowland gorillas is unknown due to their dense, remote rainforest habitat and their elusive behavior, especially around people. Without an accurate population count, it is extremely difficult for managers to conserve and protect the species or advocate for conservation policies. I'll be discussing three main techniques used for assessing Western lowland gorilla population sizes. These are camera traps, fecal and nest counts, and environmental DNA combined with nest and fecal counts. So camera traps, first of all, are usually placed at multiple locations known to be natural feeding hotspots within a gorilla territory. These cameras can be placed and left for a period of time depending on the battery life of the camera, causing minimal disturbance within the habitat. One of the main benefits of camera traps is that not only do we get to count groups, but we can also record the behavior of the species. However, the major downside is that this requires a massive amount of data processing, and it's based on the assumption that this elusive species will actually be frequenting these spots. The second technique I looked over was going out in the field and counting nest piles and dung piles. Based on gorilla behavior, it is known that gorillas will build sleeping nests and will defecate next to those nests every night. Based on this, researchers can get a rough estimate of group size. This is also minimally invasive because this is usually done after the group has left the site. However, it has been shown that gorillas will build multiple nests a night, causing overestimates. And this also requires knowledge of how long it takes the dung in the nest to decompose. Based on the size chart presented on my poster, you can get a rough estimate of age class within a group, but there's also overlapping size classes and the sex of the individual is largely unknown. The last technique I evaluated for censusing Western lowland gorillas was using environmental DNA or eDNA, along with nest counts and fecal counts. Genetic analysis has become a booming field in quantitative ecology due to decreasing costs and increasing data loading abilities. This is another passive monitoring technique that uses DNA inherent feces found in nesting sites to create genetic profiles on individual gorillas within a group. In this technique, nest counts can be verified by comparing the genetic profiles of the individuals, reducing overestimations. The sex can also be determined of individuals, giving a more clear and comprehensive analysis of Western lowland gorilla populations. Based on my finding, I recommend that researchers use a dynamic approach of counting nests and dung piles alongside using eDNA genetic profiles. This technique is minimally invasive, yet allows a more accurate censusing by reducing overestimates. Collecting accurate population census of endangered species is critical to how that species is listed and managed. With this technique, hopefully researchers will be able to get those accurate counts and be able to assess trends in future population sizes and demographics of Western lowland gorillas. So thank you for joining me on this.